Welcome
to the
back,
new era of
everyone. Ready
video
for another
creation with
deep
HeyGen.
dive?
Simply type your script to
Today,
get
we're
started.
tackling genetics, specifically inheritance and variation.
Definitely.
You guys send in some really interesting stuff, focusing on how traits get passed
Oh, a
down,
classic.
and how all this amazing variation happens. I know, right? Okay,
Yeah,
so
it's a
before
story
we
that goes
get all
way
into
back,
genes and
like
chromosomes, let's
ancient
start with a simple
civilizations back.
question. Why do kids look like their parents?
It's a question people have been asking forever, right?
Oh, like what?
I mean, even before the whole science of genetics, people were using that
Right.
knowledge. Like, think about how people used to breed crops and animals. You know, our sources actually talk about, like, Indian farmers
Really?
thousands of years ago.
Wow.
They were selectively breeding cows to get specific traits. Yeah, like they created
That's
breeds like the
incredible, like just
Sahiwal cow.
observing and
They're basically
selecting.
manipulating
But then, of
genetics
course,
without even knowing
along
what
comes
genes
Gregor
were.
Mendel, our pea plant pioneer.
Exactly.
Totally. So Mendel,
He
mid-1800s,
really brought science into the picture,
revolutionized
huh?
how we understand inheritance with his pea plants.
And what's amazing is, like, how systematic he was.
Yeah. So
Controlled
could we dive
crosses,
into one of
statistical
his
analysis,
experiments?
Right.
The
multiple
tall and
generations
dwarf
of plants. It was
pea
real
plant
science.
one.
Yeah, a classic. So he started with these
Gotcha.
true breeding plants, right?
Right, what?
Meaning they always produce the same trait, generation after generation. So he crossed a true breeding tall plant with a true breeding
Wait,
dwarf
so
plant.
even though
And
one
guess
parent
what?
was short, all the kids
All
were
the
tall?
offspring, the F1 generation, were tall.
Right, right.
Yep. That's where the whole dominant and recessive thing
Okay.
comes in. Mendel figured that traits are controlled by these factors, we call them genes now, and they come in pairs, one from each parent.
Makes sense.
So in this case,
Okay, I'm
the
following.
tall factor, let's call it T, is dominant over the dwarf factor. So even if a plant gets a T
So
from
the
one
T isn't
parent
gone, it's
and a
just
T from the
hidden.
other, it'll still be tall because T kind of
Sneaky.
overpowers T.
So-
Exactly, lurking in the background. And here's the cool part. When Mendel self-pollinated those
No
tall
way.
F1 plants, the dwarf trait popped up
So
again
the T
in the
was
next
just
generation,
waiting for its
the
chance,
F2. About a quarter of the F2 plants were dwarfs.
which basically means
Exactly. This is what led to Mendel's law of segregation. It means
So
that
they
when you
split
make
up.
gametes, sperm and egg, the two
Oh, I see.
factors or
So those
alleles
F1 plants
separate.
with both a T and a T
Exactly.
made
Each gamete
gametes,
only gets
and
one.
half of them had a T and half had a T.
Right.
And that's how you get that classic 3-to-1 ratio.
And then those gametes, they combine randomly during fertilization.
Wow, Mendel was really
Bingo.
onto something. It's like he
3
figured out the
tall
code of
plants for every
inheritance.
one dwarf plant.
Mind-blowing.
That,
Okay, before we
I
go further,
know,
can we make
and
sure we're
all
all on
without
the
knowing
same
about
page with some of the
DNA or any of
lingo?
that.
Like genotype and phenotype?
Sure. Oh, yeah, those are important. Genotype is the actual genetic makeup, the combination of alleles an organism has.
Got it.
So, like T, 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 T,
So
T, T,
genotype
those
is
are
the code,
genotypes.
and phenotype is
But
what
what you see, like the actual trait,
I like it.
that's the phenotype.
Okay, so for our pea plants, TT and T both have
Perfect
the tall
analogy.
phenotype. Only TT gives you the dwarf phenotype. Okay, that makes sense.
Right,
But wait,
because of
is
that
there a way to
dominant T.
figure out the genotype of a plant if it has a
Exactly.
dominant phenotype? Like if it's tall, how can you tell if it's TT or TT? Oh, tell me more.
Ah, good question. Mendel came up with this clever trick called a test cross. You cross the plant with a dominant
So
phenotype, in this
TT
case,
for our
tall,
pea plants.
with a homozygous recessive individual.
That's so cool.
Exactly.
Like
and then
a
by
genetic
looking at the
detective
offspring, you
game.
can figure out the
Okay,
genotype
so we've got
of the
Mendel's
original plant.
laws, dominant and recessive alleles, genotypes, phenotypes, even test crosses. I'm feeling pretty good about Mendelian genetics so far. But it can't always be this simple, right? I knew it.
You're right, it gets even more
what
interesting.
then?
Like, what happens when things don't follow this neat dominant recessive pattern?
Ooh, fancy words. What are
Well,
those?
our sources talk about incomplete dominance and codominance.
Okay, let's break it down. What's
They're
incomplete
inheritance
dominant?
patterns that are a bit more, well, nuanced. Think of it like blending traits.
Hmm.
A classic example is the snapdragon flower. If
Uh-huh.
you cross a true breeding red
Pink.
flower
What?
with a true breeding white flower, RRR, you get pink offspring.
Okay, so it's like they're both
Get
kind of
pink.
showing up.
So in this case, neither red nor white is fully dominant. Exactly. The heterozygote with one R and one RR has a phenotype that's right in between
That's
the two
so
parents.
cool.
Like
So
the
it's
red
not
and
a
white
case of
are
one
both
allele
sort
like
of partially
completely
expressed,
blocking the other
which
one.
makes pink.
Okay, now what's codominance?
Nope, they both contribute.
Okay, I need an example for this
Codominance
one.
is where both alleles are expressed equally and separately in the heterozygote. The human ABO blood group system is a great example. Remember IA, IB, and I.
Kind of.
So IA and IB, those are codominant. If a person inherits IA from one parent and IB from the other, they'll have both A and B antigens on their red blood cells, giving them blood type AB.
Ah, so it's not a mix.
Both
it's like
alleles
both
are
alleles
expressed
are saying,
fully,
hey,
not
we're both
blended
here.
like in incomplete dominance.
Okay, so
Exactly.
it gets a bit
And
more
it also
complex
shows the
than
concept
simple
of multiple
Mendelian
alleles.
trait.
You have more than just two options for that gene.
How so?
For sure. And this reminds me, population studies are really important in genetics. You might not see
Right,
all
that
the
makes sense.
possible
Okay,
alleles
are
in
you
just
ready to
one
go
family.
even deeper?
You need to look at bigger
Good.
populations.
Because Mendel didn't just study one trait at a time, he also did dihybrid
Always.
crosses. Exactly. So what happened when he did that?
Ah, yes. Two traits at once.
So like yellow and round versus green and wrinkled.
Well, he looked at seed color and shape, for example.
All yellow and round, right? Dominant traits.
Exactly. And you can probably guess what happened in the F1 generation.
Those recessive traits come back?
You got it. But then in
What
the
ratio?
F2 generation,
Whoa!
they did.
Break that down
And in
for
a
me.
very specific ratio. 9 to 3 to 3 to 1.
Wow, okay.
Okay, so 9 yellow
And this
round,
led to
3 yellow
another of
wrinkled,
Mendel's
3
laws,
green
right?
round, and 1 green wrinkled. It did. The law of independent assortment. It basically states that the alleles for one gene, like seed color,
So like
separate
flipping
independently
two
of the
coins
alleles
at the
for
same
another
time.
gene, like seed shape, during gamete formation.
That's why you get all those different
Perfect analogy.
combinations in the F2
The
generation.
outcome of one flip
It's amazing how
doesn't affect
Mendel
the other.
figured all this out, without even knowing about chromosomes or anything.
Precisely.
Oh, I'm ready. Bring on the chromosome.
It's truly groundbreaking. But now we're going to move beyond Mendel.
Sadly, yes.
Okay, so remember, Mendel's work was kind of ignored for a while. But then in the early 1900s, his ideas were rediscovered. And at
So
the
we're
same
finally
time,
connecting those
microscopes were
abstract
getting better,
factors to something
and scientists
real.
could finally see chromosomes. Exactly. They could see chromosomes during
I'm ready
cell
for those
division, especially
aha
meiosis.
moments. What did they see?
And they saw patterns that matched Mendel's laws. Okay, first,
Okay.
they saw that chromosomes, just like Mendel's factors, come in pairs in regular cells. And during meiosis,
It's
those pairs
Mendel's law
separate.
of segregation,
Each
but
gamete
with
only
actual
gets one
chromosomes.
chromosome from each pair. Sound familiar? Exactly. And they also
Oh,
saw
so
that
that
how those
explains the
chromosome
law of
pairs
independent
line up and
assortment.
separate is
This is
totally
blowing my
random.
mind.
You got it. It's all coming together. All these observations led to the chromosomal theory of inheritance, which basically says that genes are located on chromosomes.
It's like
the
all
way
the pieces of the
chromosomes
puzzle are
behave during
finally fitting
meiosis
together. We've got Mendel's
explains how
laws, we've
traits
got
get
the
passed
physical
down.
basis of inheritance with chromosomes, and all of this set the stage for like this explosion of genetic research that came after.
And it's still going strong
Fruit
today.
flies. What's
But
so
before
special
we
about
move
them?
on, I want to mention Thomas Hunt Morgan. He did a lot of work with fruit flies.
How so?
They might seem simple, but they're amazing for genetics research. They're small, easy to breed, have a short generation
So,
time,
what did
and
Morgan
they show a lot
discover
of
with his
easily
flies?
observable traits. Perfect for studying inheritance. Well, one of his big findings was about linkage.
So they don't
He
always
found that
assort
genes
independently
close together
like
on the
Mendel
same
observed?
chromosome tend to be inherited together. Not always. He found that the closer two genes
Makes
are on a
sense.
chromosome,
But
the more
could
likely
linked
they are to
genes
stick
ever be
together.
separated?
He called them linked genes. Yes. He also found that even linked genes can be separated and
What's that all
rearranged
about?
through something called recombination. It happens during meiosis. It's pretty cool. During
Whoa,
meiosis,
really?
homologous chromosomes can swap segments with each other, like shuffling
So
DNA.
even though they're physically linked,
Yeah, it
they can
creates new
still get mixed
combinations of
up.
alleles, even for genes that are linked.
I bet that
Exactly.
made
And that's a
mapping
major
genes on
source of genetic
chromosomes even
variation.
more
It's like
tricky.
nature is always shuffling the deck. It did, but it also made it more informative. One of Morgan's students, Alfred Sturtevant, figured out how to use
So,
the
like, the
frequency
more
of
often they were
recombination
separated, the
to
farther
figure out
apart
how
they
far
were.
apart genes were on a chromosome.
That's incredible. Mapping the relative positions of
Exactly.
genes
And this
based
led
on
to the
recombination.
first genetic maps. It was a huge breakthrough.
Right,
But
right.
we have to remember, not all traits follow those neat Mendelian
Polygenic
patterns we've been
inheritance.
talking about.
Sounds
Like
complicated.
polygenic inheritance and pleiotropy. Single trait.
Instead of just one gene, one trait.
It basically means
Okay, give me
multiple
an example,
genes
something relatable.
influencing a... Exactly. Human skin color is a classic example. It's not just light or dark.
So each
right?
gene adds a
There's
bit
a
of
whole
color.
spectrum because multiple genes contribute to how much pigment is
Ah,
produced.
so it's genes and environment.
Exactly.
Nature and nurture.
And things like sun exposure also play a role.
Meaning what?
Both important. And then there's pleiotropy, which is kind of the opposite of polygenic inheritance.
Wait, how is that
A
possible?
single gene can influence multiple, seemingly unrelated traits.
Okay, give me an
It's like
example.
a gene having a ripple effect. It might code for a protein involved in lots of different things. Phenylketonuria,
Right.
or PKU.
Hmm?
It's a genetic disorder from a mutation in a single gene. If that gene isn't working right,
Wow,
it can
so
cause
one
intellectual
gene can
disabilities,
impact brain development,
but
hair
it also
color, and
affects
skin color.
hair and skin color.
This is making me think about the whole nature versus nurture
Exactly.
debate.
It
again.
highlights
Seems
how
like it's both,
interconnected
right?
our genes are.
Okay,
It
we've
is.
covered a
Genes
lot.
provide a
Mendel's
blueprint,
P's,
but our
chromosomes,
environment and
linkage,
choices influence how
recombination,
that blueprint plays
polygenic
out.
inheritance, pleiotropy. Wow. It's amazing how much we know about inheritance, but we can't forget about how sex is determined, right? That's another layer of complexity.
Absolutely. And it wasn't until
Insects.
scientists
Okay,
started
tell me
studying
more.
insects that they began to understand how sex is determined genetically.
Okay.
Back in the late 1800s, this scientist, Hanking, was studying sperm development in insects. He noticed this strange structure in some cells. He called it the X body. Turns out it was the X chromosome, but he didn't know
Interesting.
that at
So
the time.
that was a
And
clue
what was
that
weird
this
was that
chromosome
only
might
half
be
of the sperm
involved
cells
in
had this
determining
X
sex.
body. Exactly. Further research led to the understanding of the X-Ophects determination system in some insects.
So.
Females have two X chromosomes.
So it's like
XX.
a 50 to 50
Well, males
chance.
have
Two
just
X's
one
your
X,
female,
XO.
one X
So
your
that
male.
second X is what makes the difference.
Females X, X males XY.
It's
But
a
why
good way to
is
think
that
about it.
Y
But of course, we
chromosome
humans
so
have the
important for
XY
being
system.
male? Right.
It carries genes that kick
So
off
for
the
humans,
development
it comes
of male
down to
characteristics
which sperm
during
fertilizes
early
the egg.
development, like a master switch.
And that's totally random.
Exactly. An X-bearing
Okay, that
sperm
clears up some
or a
old-fashioned
Y-bearing
ideas
sperm.
about one parent being responsible
Totally
for the
random,
sex of the
50-50
child.
chance.
I like that. We also have to mention that unusual sex determination system
Yeah, it's a
found
team
in
effort,
honeybees.
genetically speaking.
Refresh my memory. How does that one work?
Oh, yeah, the haplodiploid system.
Oh.
It's super interesting. So
Okay.
females develop from fertilized eggs, just like us. They're diploid,
Whoa.
two sets of chromosomes. But males,
So the
they
males are
develop
basically
from
clones
unfertilized
of the
eggs.
queen. What
They're
the?
haploid, just one set of chromosomes.
Yeah.
In a way, yes,
I can
they
imagine.
inherit everything from her. And this leads to some strange family dynamics. Males don't
That's
have fathers,
wild.
and
It
they
just
can't have
shows you how
sons.
diverse
But they
sex
do have
determination
grandfathers
can
and
be
can
in
have
nature.
grandsons. Nature is full of surprises. But now, as we wrap up this part of our
Mutations,
deep
right, those
dive,
changes in the
we
DNA
have to remember that
sequence.
genes aren't static.
And I'm
Exactly.
guessing those
Mutations are the
changes can have a
ultimate source
huge
of genetic
range of
variation.
effects.
They can be small or large.
That brings us to the topic
Absolutely.
of
From
genetic
being
disorders.
totally harmless to causing serious genetic disorders. It
Okay,
does.
what's the difference?
And we can generally categorize them as either Mendelian disorders or chromosomal disorders. Mendelian disorders are caused by mutations in single genes.
Okay, that makes sense.
And
I've
chromosomal
also heard
disorders are
about
caused by
pedigree
problems with
analysis
the
being used
number
to study
or
these
structure of
disorders.
chromosomes.
What's that all about? Ooh, I like that.
Yeah, it's like a genetic detective tool. You trace a trait or
Like a
disorder
family
through
tree,
a family,
but with the genetic
using
focus.
symbols to represent everyone and their relationships. Exactly. And
So,
by
like,
analyzing
is it
those
dominant or
pedigrees,
recessive?
geneticists can figure out how a trait is inherited.
It's like piecing together a puzzle.
Right. And they can figure out the chances of someone inheriting a disorder.
This has been quite a journey.
And that
Mendel's
information is super
P's,
important for
chromosomes,
genetic counseling
linkage,
and family planning.
recombination, all these different inheritance patterns. Totally. And it lays the groundwork for so much more.
It's a lot to take in, but it's fascinating, isn't it? It does. In the next part, we'll look at specific examples of genetic disorders and how all this knowledge is used in the real world.
Welcome back to our genetics deep dive. We've got the basics down, Mendel's laws, chromosomes, all that. But I'm really curious about how all of this plays out in real life. Right. Like when there's a glitch in the code.
Well, one way is through genetic disorders.
Okay, like what?
Exactly, mutations. And our sources have some really interesting examples.
Sex-linked, so on the
Color
X
blindness
chromosome.
is a classic one. It's a sex-linked recessive disorder.
So that's why it's way more
and
common
recessive
in
means
men.
you need two copies of that messed up allele to actually have the
trait. If they get
trait.
that one bad copy, boom, color blindness.
Because they only have one X chromosome, both parents.
Which is less likely to happen.
Yep. But women need to inherit it from...
Wow.
Much less likely. Something like 8% of males are colorblind.
It really shows how those sex-linked inheritance patterns can
females.
make some traits more common in one sex. Oh, yeah, hemophilia.
Absolutely.
I've heard of that. But remind me, what is it exactly?
Speaking of sex-linked recessive disorders, there's hemophilia. It's a bleeding disorder. Basically, there's a mutation in a gene.
So like one little
on the
protein
X
gone wrong can
and
cause
it messes
all these
up
problems.
one of the proteins needed for blood clotting.
And historically, that could be really dangerous, right?
Even a small cut can bleed for a really long time.
Oh, yeah, I've heard about that.
Definitely.
Hemophilia spread through European royalty, right?
Queen Victoria and her family.
Had one normal copy of the gene and one mutated copy.
Exactly. Queen Victoria was a carrier.
So she... And some of them
she
were
didn't
royalty,
have hemophilia
so
herself,
it spread through the royal family.
But she could pass on that mutated copy to her kids.
Wow. Okay, let's shift gears a bit.
This is like a real-life genetics lesson playing out across history.
I know that's genetic, but it's not on a sex chromosome, right?
Right. Sickle cell is autosomal recessive.
Meaning?
The gene is on one of the non-sex chromosomes. So you need two copies of the bad allele to have the disease.
So you could still have some
symptoms. That's interesting. You might not have sickle cell anemia, but you could have sickle cell trait.
Interesting. It's not just a simple, like,
you
have
might,
it or you
especially
don't.
under certain conditions, like if there's low oxygen. Nope, there can be a range.
Okay, what is it?
But what's really fascinating about sickle cell is the specific mutation that causes it.
Hemoglobin, the thing that carries oxygen in red blood cells.
In the gene for hemoglobin. That's the one. And just that one tiny change,
What causes all that?
It changes the hemoglobin molecule, which can make the red blood cells deform into a sickle shape.
It's crazy how one tiny change in the DNA can have these huge ripple effects throughout the body.
It really highlights how important even the smallest parts of our genetic code are.
We talked about
And
PKU
speaking of
before,
single
the
gene
one that affects
mutations, there's
multiple traits.
PKU, phenylketonuria, right, pleiotropy, and it's
So if
caused by a
that enzyme
mutation
isn't
in a
working,
gene, phenylalanine, for an enzyme that breaks down an amino acid called phenylalanine,
that's why
builds
they
up
screen
and
newborns for
can be
PKU,
toxic to
right?
the brain, leading to developmental problems. Exactly.
So it's not
Early
always a
detection is
case of
crucial.
like,
The
oh, there's
good
a
news is
genetic
that it can be
problem, nothing
managed
you
with a
can
special
do.
diet.
What about thalassemia? That's another genetic blood disorder,
Sometimes
right?
knowing the cause can help us find ways to manage it. It is, and it's
Okay,
also
how so?
autosomal recessive. It affects hemoglobin, but in a different way than sickle cell. In
Interesting.
thalassemia,
So too
it's not
little
that the
hemoglobin,
hemoglobin
but
is
it's normal
abnormal,
hemoglobin,
it's that there's just not enough of it.
like alpha thalassemia,
Right.
beta
And there are
thalassemia.
different types depending on which part of the hemoglobin molecule is affected. Exactly. And the severity can vary depending on how many genes
It's
are
amazing how
mutated.
complex these things
Some
can
people
be.
might just have mild
We've talked about
anemia,
single
others
gene
might need regular
disorders,
blood
but what about
transfusions.
those chromosomal disorders? The ones where there's a problem with the number or structure of the chromosome. Right. Trisomy 21, there's an extra
Oh
copy
yeah,
of
those are
chromosome
fascinating.
21.
Down syndrome is probably the most well known. Exactly.
It's important
And
to
that extra
remember that people
chromosome
with Down
leads to
syndrome
those
are all
characteristic features
individuals,
and
each with their
developmental
own
differences.
strengths and challenges. What causes that one?
Absolutely. And then there's Klinefelter syndrome, which affects
Interesting.
males,
What happens because of that
an extra X chromosome.
Yeah.
So XXY instead of XY. It
Okay.
can lead to
And there's
things
also
like being taller
Turner
than
syndrome.
average, some feminine characteristics, and often infertility.
Right. Wow.
Right, that
So
one
too
affects
much
females,
or too
and it's
little
actually a
genetic
missing X
material can
chromosome,
cause
so just one X
problems.
instead of two.
Before we move on, I think we should talk about the
It's all
ethical side
about
of things.
balance. And these chromosome disorders really show that.
It's amazing how much we can learn from
Oh
these
yeah,
tests.
for sure. Especially with genetic testing becoming more common. Right. But it also brings up questions about
It's a
who
lot
should
to
have
think
access
about.
to that information and what it means for privacy and potential discrimination. And what about prenatal
It raises
testing?
questions about
We
reproductive
can screen for
choices
genetic
and the
disorders
possibility
before
of
a baby
selective
is even
abortions.
born.
But that it's like with all this
It's
knowledge
a
comes
complex
great
issue,
responsibility.
and we need to be careful about how we use this technology. Couldn't have said it better myself. So we've covered
It's a
some pretty
lot,
heavy
but
stuff.
it's
Single
also
gene disorders,
fascinating. I'm
chromosomal
ready to see where
disorders,
this goes
the
next.
ethical considerations.
Oh, okay, like what's the bigger picture?
Well, in the final part of our deep dive, we'll zoom out a bit. Exactly. We'll look at
Sounds
how this
good. I
genetic
can't
knowledge
wait.
is being used in healthcare and
All
how
right,
it's
welcome
changing
back
how
for
we
the
think
final
about
part
human
of our
evolution.
genetics deep dive. It's been quite a journey, from those pea plants to all those complex human disorders. Wow.
Yeah,
I like
it's
it.
been
Big
amazing.
picture thinking. So where
But
do
now
we
I
start?
think it's time to broaden our perspective a bit.
Oh, absolutely. I'm hearing more and more about personalized medicine,
Well,
genetic
genetics is playing a huge
testing,
role in
all
healthcare
that.
these days. It's all connected. Understanding inheritance and variation, it's not
Totally.
just
So genetic testing,
theoretical
is that
stuff.
becoming
It
more
has a
common?
huge impact on how we deal with diseases.
What kind of information can you actually get from those
It is.
tests?
It's getting more accessible and affordable, which is great. You can find out if you're at risk for certain diseases, and doctors can
It's like
use that
peeking
info
into
to
your own
personalize
instruction
treatment plans.
manual.
Plus, we can screen
seeing
newborns
what's
for genetic
potentially in
disorders.
store for your health. Right. It's not like a fortune
It
teller or
is.
anything.
But we have to remember, genes aren't destiny.
And there's still so much we don't know about how
Exactly. They
genes
give
work
you
together and how
information,
the environment
but
affects
it's not a
them.
guarantee.
Epigenetics. I've heard that word, but I'm not sure I get it.
Absolutely. And that's where epigenetics comes in. Think of it like this. Genes can be turned on or off, or like their activity can be increased or decreased based on things
Wow,
like
so it's
your
not just about the
lifestyle,
genes
your
you inherit.
diet, stress, even exposure to toxins.
That's so interesting. So, like, even if you have a certain
Nope.
gene, it
It's
might
also
not
about the
actually
choices
do
you make and
anything
the world
unless
around you.
something triggers it.
Yeah.
And that changes how we think about healthcare, right?
It's a simplified way to think about it, but for sure. It's not just about treating
I like that, a
diseases.
more holistic
It's about
approach.
preventing them in
It's
the first place
empowering. It
by
feels like you have
promoting
some
healthy gene
control, not
expression.
just at the mercy of your genes. It's not just
Exactly. We're
genes, it's
starting
the
to see healthcare
environment, it's
in a
our
whole new light,
choices, it's like a
considering
whole
how everything
system.
is connected.
Always.
Absolutely. Okay, ready to switch gears again? What about
Oh, yeah,
genetics
like those DNA
and
tests that tell you
human
where your
evolution?
ancestors came from.
Ancestry and all that.
It's like a genetic time machine.
Exactly. It's revolutionizing how we look at our past, both personal and as a species. It is. We can trace our
What are we
lineage,
learning from
find
these
relatives
tests?
we didn't know
Any big
about, and
surprises?
even see how ancient populations moved around the globe. Well,
What?
for
So, like,
example,
we're
we
not
found
as
out
different
that
from them as we
modern
thought?
humans interbred with Neanderthals and Denisovans. Yeah,
That's wild.
we carry a bit of their DNA within us. It's changing
It's
how we
like
think about
the more
human
we learn about
diversity in our
genetics, the more
evolutionary
we
history.
realize how
We're all
interconnected we
connected,
all
way
are.
more than we realized.
I know, it's
Absolutely.
exciting to
And
think
as
about.
we keep unlocking the secrets of the genome, who knows what else we'll
Wow,
discover.
that went by fast. We've covered so
And on
much.
that note,
Mendel's
I
laws,
think we've
inheritance
reached the end of
patterns,
our deep dive.
sex determination, genetic disorders, how it all affects healthcare, human evolution.
It's been quite a ride.
To our listeners, thank you for joining us on this deep dive into the incredible world of genetics. We hope you've enjoyed it. We hope it sparked your own curiosity, and maybe even inspired you to keep learning about this amazing field. There are still mysteries
Because
to
there's still
solve,
so much
new
more to
things to
discover.
find
The
out,
story
and
of
those
genetics
ethical
is
questions,
far
we
from
have to
over.
keep talking about those.
And remember, this
So
is
keep
about all
those
of
minds
us.
curious,
It's
keep
about who
asking
we are,
questions,
where
and
we
keep
came
searching
from,
for
and what the
knowledge.
future
Until our
holds.
next deep dive, keep exploring.